So thank you very much. And thank you to the Neurological Foundation for giving me this opportunity to talk about my work. So I'm just going to give a very brief background on me. So I completed my PhD at the Center for Brain Research, where I was investigating the biology of the blood-brain barrier. There, I was lucky to have access to human brain tissue collected either from surgical resections from epilepsy patients or from postmortem brain tissue. While we used monolayers, so some of these images are monolayers of the cells that we used. We also used explants, so 3D pieces of tissue from either the meninges, so the thin layer of tissue that covers the brain, or from the choroid plexus, which sits inside the ventricle and makes cerebral spinal fluid, or CSF. It was around this time that I'd finished my PhD, and serendipity happened by way of several things coming together to happen all at once. So the first one was one of my colleagues recommended this book to me by Matthew Walker called Why We Sleep. In case you're wondering, no, I'm not a sales rep for Matthew Walker. I was just fascinated by this book, and it was amazing to know about all the things that I don't know about sleep and what it does and why it's important. At the same time, I also watched a TED Talk by someone named Jeffrey Iliff, and he was talking about the discovery of a new thing that happened in the brain, which involves CSF clearing things from the brain and potentially staving off Alzheimer's disease. And additionally, and lastly, at the same time, I was also, like I said, studying the choroid plexus. And in doing so, I took a picture of this tissue, and this is a piece of choroid plexus tissue from a postmortem human brain. And I just fell in love. I thought this is the most beautiful thing I've ever seen, and I need to learn more about it. So I got in touch with Professor Iliff, and I was awarded a postdoctoral fellowship to go study in lab. So there I was able to learn about sleep and CSF dynamics, but also to ask specific questions about the role of the choroid plexus in this physiology. And this is what I'm going to tell you about today. If my mouse works, there we go. So I included this image because I think it's a really great visual of what I'm going to be talking about. And so if nothing else, I'd like you to at least keep this image in your brain forever and let it motivate you to at least get a better night's rest. So sleep. All animals sleep, down to even worms. And while during sleep, animals don't eat, they don't reproduce, they don't forage, and are especially vulnerable to predation. Yet the persistence of sleep, despite evolutionary pressures and the deleterious effects of sleep deprivation, indicate that sleep serves a critical function or functions that cannot be easily bypassed. Now, regulation of sleep can be most simply described using a two-process model, whereby sleep occurs from a combination of sleep pressure, referred to as process S, which results from increasing time awake, and circadian rhythms, referred to as process C. So while sleep pressure builds up, it can only be dissipated by sleep itself. Alternatively, the circadian rhythm is the body's internal clock and is driven by light cycles or exposure to the sun, which is approximately 24 hours. It is mainly regulated by melatonin released from the pineal gland, but it does reset itself every 24 hours. On the other hand, sleep itself, like I said before, can only be dissipated. Sleep pressure can only be dissipated by sleep. So this explains some of the reasoning why if you have a really bad night of sleep, or if you don't sleep for a full night, the next day, while you're very tired, you still feel very alert. So this is a combination of those two processes working together. Now, sleep physiology itself is something that I would like to discuss. So sleep characterization is usually happens using something called polysomnography. So polysomnography is a comprehensive test that involves many different measures. So it's measuring brain activity with EEG or electroencephalography. It also measures eye movements, muscle activity, skeletal muscle activation, heart rhythm, and all those combined allow scientists and researchers to actually characterize the different stages of sleep. So if you can see here, this picture on the right, So we've got different stages of sleep. And if you can see here, wakefulness compared to REM or rapid eye movement sleep are very similar and are characterized by low amplitude, high frequency EEG waves. The main difference between REM and the awake state is actually the reduction in muscle tone, something called muscle atonia. Then the other stages of sleep, also referred to as non-REM, involve differences in the frequency and amplitude of the brainwaves that can be measured. So as you progress from stage 1 to 2 and into 3, there's actually an increase in the amplitude and a decrease in the frequency of these brain waves. And this is thought to be due to the synchronous activity of neurons within the brain. So normal sleep cycles, so during the course of an evening, 7 to 9 hours of sleep, a person normally will cycle through several sleep cycles that are about 90 minutes each. And this is shown here in this diagram, on the left. If you can see, the early stages of sleep are mostly dominated by these non-REM states, or mostly N3, which is the most slow wave sleep. Whereas in the later parts of the evening, or getting into the early morning before you wake up, it's dominated by REM sleep, and that's usually where dreams occur. And a lot of the reason why sometimes you can remember your dreams is because a lot of them occurred right before you woke up. So sleep and circadian dysfunction have long been symptomatic hallmarks of various neurological conditions, including Alzheimer's disease. So accumulating evidence now suggests that sleep and circadian disruption could occur very early in the course of disease, and not only as a result of disease processes, but may also contribute to disease progression. So a few examples of where this might occur is something called rapid eye movement behavior disorder, in which the normal paralysis, muscle paralysis or muscle atonia, which occurs during REM sleep, is lost. And actually up to 80% of people that suffer this condition go on to develop Parkinson's disease or something similar, alpha-synucleinopathy. So it's when the protein within Parkinson's actually accumulates. Another example of this is chronic poor sleep quality or poor sleep health. Can be linked to the protein kinetics of amyloid beta. So amyloid beta is a protein that accumulates and is found in the brains of people with Alzheimer's disease. And a study showed that in young, healthy adults, a single night of sleep deprivation is enough to increase the amount of amyloid within the brain. And then lastly, the other thing I want to bring your attention to, like Sarah mentioned, is the involvement of sleep disruption and circadian dysrhythmia in the context of critical illness. So while medical advancements mean that a lot of people that are critically ill, actually are able to survive and be released from hospital, up to 80% of these people actually go on to develop some form of cognitive decline. And there's a few studies now that suggest that there's a strong link and relationship between the sleep fragmentation and circadian disruption that they experience during the ICU environment that could potentially drive the cognitive decline that's seen later. So this is the general biology that I'm interested in. So I'd like to, so I basically want to be able to identify the link between sleep and circadian disruption in neurological disorder. So it's a big ask, but one thing that I'm particularly focused on and being this potential link is CSF physiology. So CSF, or cerebrospinal fluid, historically is mostly known to be acting as a cushion. So it cushions the brain and protects it. But it's also something that's in constant movement. This is something that we've known for quite a while, actually, that CSF that's secreted from the choroid plexus within the ventricles actually moves down through these foramens and then moves around the outside of the brain, something called the subarachnoid space. But actually, more recently, we have come to understand that it doesn't just move around the outside of the brain, that CSF is actually able to penetrate the brain tissue exchange with the interstitial fluid. So that's the fluid that is around the brain cells, and then can be cleared into lymph nodes, and is a mechanism for clearing some of the metabolites that are produced by neurons just throughout their normal activity. So this physiology is something that is now referred to as the glymphatic system. So the glymphatic system itself describes the movement, exchange, and clearance of brain solutes through perivascular pathways that surround brain vessels. So if you follow my mouse here, so it's essentially the movement of CSF in the subarachnoid space down through beside these penetrating vessels. It's allowed to exchange through which is facilitated by astrocyte end feet. So it exchanges within the brain tissue and then can be cleared along these perivenous pathways. So, and then eventually ending up into the lymph nodes and the blood. So because of this close physical or anatomical approximation to the vasculature, glymphatic function, so the actual movement of CSF, is influenced by factors that are related to vascular function, such as arterial pulsation, vasomotion, and sympathetic tone, but can also be impacted by certain vascular diseases such as hypertension. So early studies that aimed to characterize the glymphatic system actually used animal models. So I'm just going to describe one of the earlier studies here. It involved the injection of tracers. So in this case, this was an MRI study, and the tracer that was used was a contrast agent, so something that is easily visualized using MRI. It was injected into the cisterna magna, so it's just a small CSF space that's below the cerebellum. And then the animal was imaged for a course of 2 hours. And what was seen was actually, I'm going to show you the movie quick and then I'll slow it down. But what is seen is the movement of this tracer throughout the brain tissue. So if you can see, I'm just going to try to slow it down here. So what you can see here is at the base where the tracer is injected into the cisterna magna, and then slowly it will go through the ventral side of the brain and then up towards the front of the brain. But what's really cool, what I want you to pay attention to, is right about here. So if you can see on the side, there's these lines where the tracer is going, and that's actually because it's following the vessels that run up alongside the brain and then eventually penetrate into the brain tissue. So this is just offering first hints of like what's exactly happening within the CSF, how it's moving and where it's going. But I told you that I was going to talk to you about sleep. So the really cool thing is that this process of CSF movement, exchange, and clearance is predominantly active during sleep. And the way that this was identified was using a similar experiment. So the injection of a tracer, but this time not a contrast agent. This time it was a fluorescent tracer, and it was imaged with a fluorescent microscope and a cranial window that was mounted on the skull so that you can see into the brain. And so this is a 3D rendering of a portion of that tissue just on the surface of the brain, with the brain vessels, the blood vessels in blue. And what I hope you can see here, so the green is the amount of tracer, the movement of the tracer kind of into the brain tissue while the animal was asleep. Whereas the yellow is the movement into the brain tissue while the animal was awake. So I hope that you can appreciate the gross difference in the ability of this tracer to move into the brain tissue from the CSF space when the animal is sleeping versus when it's awake. And then something that's really important to note actually is they also conducted studies using an anesthesia that is able to mimic slow wave sleep specifically. So this anesthesia that acts like slow wave sleep is able to reproduce very similar effects to that seen when the animal is asleep. So this suggests that specifically slow wave sleep is able to encourage or drive this process of CSF exchange within the brain tissue. Right. And so in addition to sleep, Glymphatic function or glymphatic activity is also regulated by circadian rhythms. So this was shown doing experiments, again, very similar to before, injection of fluorescent tracers and then imaging the movement of these tracers into the brain over time. And in this case, it was done while the animals were under the exact same anesthesia conditions, but just at different times of day. And what you can see here in this top panel is that during the day, so in animals, mice that are nocturnal animals, this is their normal resting period. During the day, you see an increase in this tracer movement and penetration into the tissue, whereas during the night, this is significantly reduced. So that's all well and good, and it's really cool, but it's in mice. So does this thing happen in humans? Well, it turns out that it actually does. So a group in Oslo has done almost all of the experiments regarding glymphatic function in humans. They did it sort of the same idea as what has been done in mice, but this is using contrast agent and MRI, and instead of injected into the cisterna magna, that's actually injected intrathecally, so into the spinal column, so like a lumbar puncture. And what you can see here, so I'm hoping this video will work as well, is over time, there is a movement of this contrast agent or tracer throughout the brain. And you can see it follows a very similar pattern to what I showed you in the animals. And then subsequently, the same group actually did a very similar experiment, the same thing, but the important difference here is they divided the subjects into two groups. Each group had the injection of the contrast agent into the spinal column, and then one group was allowed to sleep the whole night like normal. The other group had to stay awake the whole night, and then they imaged them both the next day. And what you can see here down at this bottom panel, so this is the difference between the subjects that slept and the subjects that didn't. So showing that the ones that slept actually had more contrast agent that remained in their brain the next day because of that loss of sleep. So this taken together is potentially suggesting that glymphatic function or impairment of glymphatic function that is maybe seen with sleep disruption or sleep fragmentation could be contributing to some of the things that we see in neurological diseases, such as the accumulation of toxic proteins. So proteins such as amyloid beta and tau that's seen in Alzheimer's disease, as well as alpha-synuclein that's seen in Parkinson's. Okay, so altogether, that was a lot and I might have talked too fast, I'm sorry. But I'd like to just summarize and hope to you that I've demonstrated that glymphatic function is essential for the clearance of brain solutes, is regulated by both sleep and circadian cycles, and potentially its reduced function in age and in this setting of disease might contribute to some disease processes. So with that said, I told you that I was going to talk about the choroid plexus, and I really want to because I'm really excited and I think it's a really cool thing. So again, just to remind you how lovely it is, this beautiful tissue. What is it exactly? So I'm going to tell you. The choroid plexus, like I said, so it's highly vascularized. It sits inside the brain ventricles. So there's 4 ventricles in the brain, and it makes the majority of the cerebral spinal fluid. And it does this through these ciliated epithelial cells. So this is sort of the CSF space, and these are these ciliated epithelial cells, and the cilia help to push the CSF into the ventricular space. And it surrounds this stromal tissue, again, that is highly vascularized. So the important thing to remember about the choroid plexus and its vasculature is that the vasculature is referred to as fenestrated. So if you don't know what that means, it just means that it's very similar to the blood vessels in the rest of our body, and that there's more of an open communication or movement from the blood into the tissue that's surrounding. Whereas the rest of the brain has something called the blood-brain barrier, and that's restricts or limits the movement of both molecules and other cells from the blood into the brain tissue. So the ability of the choroid plexus to actually sense the environment and respond to physiological conditions is heightened because of this fenestration, so this open communication between the blood and the stromal tissue. Alternatively, instead of having a really tight barrier at the blood vessels, the choroid plexus actually has tight junctions, so like things that just hold the cells really close together, located at the level of these epithelial cells. So it acts as a brain CSF barrier. So the choroid plexus does a lot of things. So it doesn't just make CSF, it does that, but it also, like I said, acts as a brain CSF barrier. It provides trophic support, so in the way of delivering nutrients and some like growth factors to the surrounding neuronal populations within the brain. And it also provides immune surveillance. So because of the fenestrated nature of the blood vessels and the ability of immune cells to be present within the stromal tissue, they're always kind of sensing the environment and can respond appropriately. So this picture here, I like to include it because I think it's a really great demonstration of how dynamic the choroid plexus is. So this is a 3D reconstruction using from MRI from a patient that has idiopathic intracranial hypertension. And so the red is the choroid plexus before, and the gray is the choroid plexus after a lumbar puncture within this patient. So this is how much the choroid plexus can just change in volume in response to a change in pressure due to a lumbar puncture, like at the very distal end of the spinal cord. Now, the choroid plexus, we don't really know a lot about its regulation, but we do know at least that it is strongly regulated by circadian rhythm. So this was recently shown by using ex vivo experiments. So taking the choroid plexus out of the tissue, putting it into a dish, and then measuring circadian-dependent gene expression. And what's really cool here is that they also, this group also showed that the choroid plexus can provide feedback, positive feedback to the suprachiasmatic nucleus. So this is the master regulator of circadian rhythms within the brain, which potentially could mean that the choroid plexus and its regulation could have far-reaching implications to the rest of the brain and brain homeostasis in general. Now, a demonstration of how circadian rhythms can regulate choroid plexus function was detected many years ago, back in the 90s, using MRI. And what these researchers did was they imaged a specific region of the brain where CSF will move, the cerebral aqueduct, and they used that as a surrogate for CSF production. And what they saw was that actually the production of CSF is diurnally or circadianly regulated. So during the nighttime, your normal resting phase, circadian production is highest, and its opposite, it's lowest during the day. And this could be a reflection of a specific circadian regulation, or it could also reflect other regulatory factors such as the presence of adrenaline or epinephrine, which is known to affect sleep and sleep cycles as well. So why do we care about the choroid plexus? And it, like, just besides the fact that it's beautiful and it's really cool and does all these important things, it's actually quite a lot of evidence so far in both animal models and in human studies have shown that there's impairment of choroid plexus function. So relating to either CSF production or increased presence of immune cells within the stromal tissue, potentially a reduction in these barrier properties demonstrated by reduced tight junction expression, and changes in the composition of the CSF. So what is actually being secreted, like changing from neurotrophic and supportive to being potentially pro-inflammatory and actually detrimental. So while these studies have demonstrated some sort of relationship with choroid plexus dysfunction and disease, it's not really clear whether the choroid plexus dysfunction that is seen is a result of existing pathology or if it's actually happening earlier and potentially contributing to disease development and disease progression. So I went to Seattle to learn about CSF dynamics in sleep, but also to ask questions about the choroid plexus and its role, not only in physiology, but also just in brain function, with the general hypothesis that conditions within the body, so sleep states, for example, circadian rhythms, maybe inflammation or injury, could then impact the choroid plexus and then in turn affecting brain function because of its ability to modulate, CSF production, immune cell presence, etc. So the main hypotheses that I had were about whether the choroid plexus was transcriptionally, so at the gene expression level, regulated by sleep or circadian rhythms, and whether this regulation was different in young animals versus old animals, and whether those changes could potentially reflect changes in function. So designed a fairly simple experiment where we collected choroid plexus tissue from young adult mice, healthy adult mice, at different times of day. So either midway through their normal active period, so that's the middle of the night, or midway through their normal resting period, which is, sorry, I might have said that wrong, but anyways, midway through their normal resting period, either if allowed to sleep or if we actually fragmented their sleep. And then we collected the tissue and submitted it for RNA sequencing so that we could get an essential profile of gene expression and how it was different between these different states. And what we actually found was partially surprising. There was actually no significant difference between gene expression of animals that were asleep versus animals that were awake, which was suggesting that at least in young healthy animals, sleep was not regulating gene expression within the choroid plexus. But we did see that there was a very strong circadian regulation, which is, again, consistent with the previous study that I mentioned. But when we looked at the actual genes that were changed in accordance with the circadian rhythm, they were mostly surrounding immune and inflammatory function. So just to give a little diagram of what looks like it's happening within the choroid plexus within circadian rhythms is like these epithelial cells that are the blue ones facing the CSF at the bottom are usually separated from immune cells. But it looks like during the resting phase of mice that there's actually an increase in some of these immune functions. So immune cells that exist in the blood, there's more activity. There's more evidence of them potentially transversing this endothelial somewhat barrier. There's also more evidence of them expressing molecules that will signal to other immune cells and get them activated. And there's also evidence of increased phagocytosis, so like engulfing potentially damaging particles or pathogens within the tissue. So that's what we see in the young mice. But then what happens in the setting of age or in certain disease conditions? So for example, when there's too much amyloid in the brain. So like I said before, there is evidence of different levels of dysfunction within the choroid plexus, and whether this is regulated circadianly was what we wanted to know. So we did the same experiment in old mice or in mice that had a whole bunch of amyloid in their brain. And what we saw was this. So please don't try to read all these words on the bottom. It's just a gene list. This is essentially just demonstrating the regulation of genes by circadian rhythm. So these are the genes that were most changed when we compare the night versus the day. And then the comparison that we did next was if we take all these genes and we look at how they're expressed in the old mice in the day versus the night. And I hope that you can see that this is incredibly different. So there's this significant loss in regulation of these specific genes when we compare old animals to young animals. And then if we do the same comparison in animals that have a whole bunch of amyloid in their brain, what we see is an even further dysregulation. So a complete loss of circadian control within this gene set, at least. So that's interesting in its own. So what's going on? We're not really sure, but what it looks like if we go back and we look at the genes that were changed in the old mice, because there were a lot, they were just different genes. So this time it looks like the function that relates to the genes that were changed is not related to immune functions or inflammatory processes, but actually ion channels and receptors. So I didn't describe this before, but just to give you an example, this is a schematic of a choroid plexus epithelial cell. And there are specific channels for like water, and there's transporters for ions like potassium and chloride and sodium. And the location of these transporters, either being at the CSF side or the stromal space side, is really important in maintaining ionic homeostasis. So the ability of the choroid plexus to control the composition of the CSF is dependent on the expression and the localization of these transporters. So it looks like for whatever reason, there's a shift in the expression of these transporters during the circadian rhythm, as opposed to immune functions. So in summary, what we see in animal models is that there's a circadian-dependent regulation that's altered with age and disease conditions. And this is in animals. So again, how does this relate to human disease? Well, we can't really do the same experiments in humans that we can do in animals, but we do know from some postmortem studies that there are changes that occur within the choroid plexus in the setting of disease that include functional things like CSF production, as well as atrophy of the epithelial cells, so when they kind of get more squished, and reduce expression of the tight junctions that link the epithelial cells together. So to try to figure out what's going on in human tissue, we took a different approach. So we did a single cell RNA-seq study. So the reason why we did that is because there are different cell types here, right? So there's epithelial cells, there's immune cells, there's cells that make up the vasculature, and there's kind of cells that reside within this stromal space. And which ones are being impacted the most by disease and potentially contributing, we weren't sure. So we did a single cell study. And I'm essentially going to breeze through this, but don't worry about it. It's very simplified. So what we did was we collected tissue through collaboration with the Neuropathology Core at the University of Washington, and with funding through the Alzheimer's Disease Research Center. We collected tissue, postmortem, fresh frozen choroid plexus tissue, isolated individual cells, and essentially labeled each one of those individual cells with a specific probe, and encapsulated in something like a liposome. And then each one of those individual cells is sequenced. And then through a lot of really fancy bioinformatics, we can look at the similar attributes of these cells and specific markers that are known to be expressed in each different cell type, and kind of cluster them and separate them based on the cell type. So we did this, and what we initially see is we get a bunch of different cell types that we expect, which is great. So we get some epithelial cells, so the cilia, ciliated ones that reside on the outside. We get some endothelial cells, so these are the ones making up the vasculature. And like I said, it's highly vascularized, so we really want to know what's happening with these cells. And then there's also an immune population that we wanted to look at. And so in summary, this is sort of busy, and I'm sorry, I'll try to get through it. What we did, so we had clinical patient data, and we have either dementia status, so people that had dementia when they passed or didn't. And we also had information on vascular disease, so whether they had severe vascular disease or not. And so we did a few comparisons to see whether there was genes that were specifically changed within these cell types when we compare the two sort of disease phenotypes. And what we see mostly is that in the different epithelial cells, that there are changes relating to inflammatory and immune responses. So remember, this is something that I told you was regulated by circadian rhythms in the young animals, as well as some cilia regulated genes. And then if we look at specifically the immune population, so because the choroid plexus has a lot of immune surveillance that's happening, in people that have dementia, it looks like the immune population has an increase or an enrichment for genes involved in protein folding, which is interesting. Also like autophagy, so cell eating, and leukocyte activation, so activation of the surrounding immune cells. And we see something mostly similar when we compare the vascular disease states. So again, just like changes in immune function and cilia, as well as immune cell activation. So taken all that together, I hope that I've been able to demonstrate that the choroid plexus has circadian-dependent regulation that changes gene expression, and this is altered in the setting of both age and disease, at least in animal models, and that there could be both central, so dementia status, or systemic vascular disease conditions that impact the choroid plexus function at the cellular level. Now, ongoing studies in the ILIF lab still happening. COVID kind of delayed everything a little bit, but we're still working closely together on a few things. So something involving longitudinal studies involving the Seattle longitudinal study to study sleep, sorry. We're doing some non-invasive MRI techniques to be able to measure glymphatic function and CSF production in at-risk populations, as well as some animal models of chronic sleep fragmentation in the context of both aging and amyloid pathology. So I didn't get into what I'm doing now too much because I just started, but I'm really excited. So I'm working with Professor Anthony Phillips and John Windsor, who are experts in critical illness, trying to understand this relationship between sleep disruption and circadian dysrhythmia with cognitive decline, with the idea that the combination of sleep and circadian disruption could be impacting both glymphatic function, so CSF movement, exchange, and clearance, but also potentially choroid plexus function, so CSF production, as well as immune and inflammatory responses within the brain. So this is the general hypothesis that we have, that acute critical illness in combination with sleep and circadian disruption could then delay or reduce glymphatic function, leading to increased inflammation and a buildup of potentially toxic solutes within the brain, leading to cognitive decline. So ultimately, we would like to be able to find drugs that can target glymphatic function specifically, encourage clearance, and hopefully prevent or reduce cognitive impairment in these patients. So that is all I have to talk about. I'm sorry if I went super fast, but I just want to say thank you very much for listening. And thank you to all of the people that I worked with, all of the people in the ILIF lab. They're all amazing. My slides keep changing. And my collaborators, as well as the Neurological Foundation for funding, the NIH, and the ADRC. So thank you very much.